Welcome to AP Biology. Today I want to talk about genetic disorders caused by separating chromosomes the wrong way. So this would happen either in meiosis or mitosis. If it happens in meiosis, that's when you are making egg or sperm. And so the homologous chromosomes will line up along the metaphase plate, and each pair will line up like this in whatever order they line up in. Independent assortment, right? And then spindle fibers will pull them to opposite sides. So nondisjunction can happen in meiosis 1. Maybe these two pairs will both go to one side over that way instead of going to each side, maybe they'll both end up going to the same place. It could also happen in meiosis 2. So in meiosis 2, it looks just like mitosis, right? It would look kind of more like this. You'd have sister chromatids that are separated out. So they're not in pairs anymore. You just have the chromosomes, the duplicated chromosomes lining up in single file. And if both of them go to one side instead of separating out, that is also nondisjunction. Can happen in mitosis as well. You might see a problem in this karyotype. So karyotype is a picture of chromosomes from largest to smallest plus the sex chromosomes. And you might notice a problem right here. So this karyotype shows that there's an extra 21st chromosome. Could have happened in meiosis 1 or meiosis 2, or actually even in mitosis of the embryo at an early stage. That could still affect the whole organism, the whole baby. Okay, so what is nondisjunction? Nondisjunction is when chromosomes separate incorrectly. So you can see here, this is in meiosis 1. Both of the chromosomes end up going to the same side. So you end up getting 3 chromosomes instead of 2. So up here, 1, 2, 3, 4. We've got a 2n number of 4. Down here, we should have an n number of 2, right? We should go from 4 chromosomes to half that much. So in other words, from diploid up here with homologous pairs that match each other to haploid right here. And instead of getting that, we don't have the haploid number of 2, we have 3, which is 1 extra. So we would call that n plus 1. Or this one is actually missing one of the chromosomes. We should have gotten 2, we only have 1, that is n minus 1. So those all resulted from nondisjunction. So daughter cells end up with the wrong number of chromosomes for that species. So this can occur if homologous chromosomes don't separate properly in meiosis 1. When that happens, you end up having a problem in all of the sperm or egg that are made by the end. They all have either too many or too few. It can also happen, though, if this is showing you that homologous chromosomes separated out just fine. They each got the haploid number of n equals 2 in this case, right? Because 2n, the diploid number equals 4 here. We've got 2n, meaning diploid. 2 full sets of chromosomes, these match, they're homologous, and these match. They separated out great, but then this in meiosis 2 doesn't separate out correctly. And when the problem is in meiosis 2, you end up making 2 cells that are just fine and then 2 cells that have a problem. Okay, so here's just an example of an AP question that you might get. This figure illustrates the X and Y chromosomes during meiosis 1 and meiosis 2 of normal spermatogenesis in mammal species. If the normal spermatogenesis is disrupted, the gametes can have different chromosomes than expected. Which of the following is the most likely cause of the 4 gametes having 2 X chromosomes and one having neither an X or a Y? Which of the following is the most likely cause of one of the 4 gametes having 2 X chromosomes? So this is showing you the normal way. It's showing that that dad has an X and a Y, and it's showing you that the X separates from the Y just like it should, and then the sister chromatids separate, and the Y chromatids separate. So this is just the normal one. So what it's asking now is what happens if one has 2 X chromosomes? So it would end up looking like this, and then one has nothing, so that would have none. So that's actually going to be a problem with meiosis 2. So this says meiosis 2, and so does that, and so does that. This one is meiosis 1. That's not true. So non-disjunction of both the X and Y. No, not the Y, because it doesn't say anything about any of the cells having a problem with the Y. It just says that one of the gametes has nothing. One of the gametes has 2 X chromosomes and one has nothing. So it's not that. Non-disjunction of the Y, no, because you'd have 2 Ys in one of the cells, so not that. So, whoops. So disjunction of the X chromosome during meiosis II is what we were looking at. If it were a problem in meiosis I, we would have all of the gametes with a problem instead of just one with too many and one with nothing. Okay, so offspring resulting from fertilization of a normal gamete with one after non-disjunction will have an abnormal chromosome number. So in other words, the sperm ended up with the right number of chromosomes. The haploid number for humans is 23, and the egg didn't. So this is n plus 1, and it's 24. So the egg has one extra chromosome. The baby ends up having one extra chromosome. We would say 2n equals, we could say 46 plus 1. We could write it that way. Or we could say it's 2n plus 1. So you do not need to memorize any of the syndromes, but here's an example of Down syndrome, the extra 21st chromosome. This is an autosome, right? All of the chromosomes that are not the sex chromosomes are autosomes. And notice that the 21st chromosome is very, very small. So that chromosome, when non-disjunction happens, the person lives usually. If you have an extra chromosome 1, though, or an extra chromosome 2, some of these bigger chromosomes, usually the baby just dies. So it's not that these chromosomes never mess up. It's just that when they do, the baby doesn't make it because there's just so much DNA. So having an extra first chromosome, usually the baby doesn't live. It's lethal. So although chromosome 21 is very small, having an extra really does change an individual's life quite a bit. Okay, so here's a picture, a graph of Down syndrome, and it correlates with age of parents. So this one is showing maternal age. So as mom gets older, she is more and more likely to have a baby with Down syndrome. And it starts to go up around 34, 35, something like that. But then it sort of spikes up a lot faster through the 40s. So this might be linked to some age-dependent abnormality in the spindle checkpoints during meiosis I, leading to non-disjunction. So this is, notice it's individuals per 1,000 births. So even if you're 39 or 40 having a baby, let's draw a straight line. It doesn't mean that you have a 4% chance of having a baby with Downs. That's 4 out of 1,000, so that's like a 0.4% chance, right? Okay, non-disjunction of other autosomes also increases with maternal age, but it's rare for infants to live for very long if you have an extra autosome because these chromosomes are just much bigger. That you can be born with an extra 13th, but I don't think you live super long with that. But you can certainly have the smaller chromosomes. Okay, infants who have one fewer or extra sex chromosomes usually do live, though, which is so strange, right? Because look at how big these X chromosomes are. So this is showing you a person who has 2 X chromosomes and a Y. Remember, typical girls have 2 X's, right? And typical boys have 1 X and 1 Y. So that right there already should probably make you think for a minute, like if you have an extra 21st chromosome, that's this, you're pretty different with just a teeny little bit of extra DNA. But here's a girl with a ton of extra DNA compared to a boy. Or the other way around, boys have an X and a Y, right? And take a look at this. The Y chromosome's tiny compared to the X. So why don't boys have very, very big problems compared to girls, or vice versa, right? And so the reason is in girls, one of the X chromosomes sort of condenses down and doesn't really get used much. It's called a bar body. And that way, girls and boys don't really have a lot of different, they don't have a very big difference in the amount of their DNA that's actually available to be used. So this is called a bar body. It's an inactive X chromosome. So unlike autosomes, a different number of sex chromosomes upsets the genetic balance less severely. It's because DNA makes RNA, and RNA makes proteins, and therefore, if you have an extra chromosome, you're going to be having too much of a protein, right? Or fewer chromosomes, you would be having too little of a protein, right? That would be a problem. But it doesn't matter quite so much with sex chromosomes because you usually shut down one of your extra X chromosomes anyway if you're a girl. So this might be, and for the Y chromosome, it might be because the Y chromosome is really small. There's really not that much on it. There's a master gene that turns on other genes that say, hey, you should develop as a boy. and a few other genes, but there's not a ton on the Y chromosome. And as far as the X chromosome, it has a lot of stuff. Like your X chromosome has tons of different genes. They have their genes for how your blood clots, right? Stuff that has nothing to do with being a boy or a girl. So everybody needs one X chromosome. If you're a girl and you have two X chromosomes, one is just inactivated as a bar body. And so this whole DNA makes RNA makes protein isn't as big of a deal because one of your extra X chromosomes is sort of condensed, sort of not used so much. Okay, so for example, if you look at a cat, this is a girl cat. She's calico, I think a beautiful cat. The gene for color on this particular type of cat is on the X chromosome, and she can be either orange or black. I think there's a I think sometimes there's a little bit of white in there too. But if you look at, but I think maybe that's in a different spot. So if you look at her genotype, she has a gene for the orange color and a gene for the black color. And that's on the X chromosome, right? And one of them gets sort of compacted down to the bar body. And so in this patch that's orange, or this patch, the cell that gave rise to those cells inactivated the chromosome that codes for black color, and it didn't make the bar body. The orange one is the one that's still used, so the bar body is the black one in this case. And then for here, any of the black spots on her, um, it was her orange X chromosome. It was the X chromosome that carries the orange color gene that became the bar body that was inactivated. And so, oops, that's the orange one that was inactivated, whereas the X chromosome with the black on it was active, and that's the one that codes for the color, the black color in cats. And kind of cool, right? So at some point in embryonic development, each cell in the female kind of randomly condenses one of the X chromosomes, and so you just have one that's used in each cell line. So the bar body is inactivated in each cell line of a female. The bar body inactivated in each cell line of a female is random, and that's the normal way that works. Okay, so here's an example of Klinefelter syndrome of a genetic abnormality where there's an extra X chromosome. So you don't have to memorize Klinefelters, but if you saw this karyotype, I would want you to see that there's a different number here. This baby or this person has XXY. So I want you to know that that's a male, because if you have a Y chromosome, you're male, but then there's an extra X chromosome. So he lives even though this chromosome is huge, right? I mean, if you have a chromosome that big, that you have an extra of, you're going to be very, very different usually. But for an X chromosome, since one of them becomes a bar body, it's pretty typical. Like the boy's not that much different. Um, so and I mean, he survives. He has normal intelligence. He's usually sterile, and he has some female traits. So it's not like completely the same as a typical male, but he can lead a pretty normal life. Again, don't memorize that it's Klinefelter's, but do notice the difference here. So most boys have 0 bar bodies because they only have one X chromosome. These boys will have 1 bar body because they have 2 X chromosomes. So they're male, they have male sex organs, they are, I think, I want to say usually sterile, they have normal intelligence. Here's a boy who has XYY. And so he has one X just like most boys, but he has 2 Y chromosomes. I would want you to be able to recognize this as a part that's different. He actually, and don't memorize this, but he actually isn't very different from other males. There's really not a lot on the Y chromosome, and he might be a little taller, but maybe some learning disabilities they think sometimes, but not a whole lot different from other males. He has trisomy X. It's a girl because there's no Y chromosome. They're just showing you that if there were, you'd have it written here. So 3 X chromosomes. So she has 2 bar bodies, and she, I think, is pretty normal. I think a little taller than average too. Here's Turner syndrome. This is a girl, but she only has 1 X chromosome instead of 2, so she will have no bar bodies. She's actually quite a bit different. Having one too few X chromosomes is actually kind of a big deal. She's a little bit more different than the other folks, which is kind of weird, isn't it? Because boys aren't that much different, and they just have that plus a Y. Anyway, okay, so a couple more definitions. So what we were talking about before, like trisomy or aneuploid, that means you have one extra chromosome or one too few chromosome. Trisomy would be one too many chromosomes. Polyploidy is a condition where you have a complete extra set. So if we go back to this, you have one fewer chromosome. You have one extra chromosome, right? But all the others are fine. They're just regular paired. With polyploidy, you have a complete extra set. So this is haploid, you have one set. Diploid, which is normal for us, you have two sets of chromosomes. If you're triploid, you have three full sets of chromosomes. And actually, there are animals and plants that are triploid or even tetraploid. They don't live with people, but I think, but they do in other organisms. So polyploidy is a condition in which an organism has more than two complete sets of chromosomes. And actually, a lot of these organisms have fewer changes than aneuploidy. Aneuploidy is what we saw with Downs, if you have an extra or one less, one fewer chromosomes. Polyploidy is when you have a full extra set, like triploid or tetraploid. So polyploid is different from aneuploidy, right? Aneuploidy is one few or extra chromosomes. Polyploidy is a whole extra set. So, like for example, strawberries have, I don't know, what do you call that? Octoploidy, maybe? So anyway, plants tolerate these genetic changes better because they can self-fertilize. So when they do meiosis, it's like an even number. Well, not necessarily an even number, but they can fertilize themselves and then get back to, when they do meiosis, get back to the number that they should have when they then go ahead and fertilize. Okay, so last thing I want to do is changes in chromosome structure. Chromosomes can break and then either be lost or added back on to either the same chromosome or a different one. So if you have deletion, that means you're missing a piece of a chromosome, and it's not one gene that's deleted, it's several genes. It's a whole section of a chromosome. So if you look at this chromosome, if this part is deleted, it looks like that. It will be missing certain genes. Duplication is when you have an extra. So maybe it came from like a crossover event where one piece came over, but the other piece didn't go to the first one. So it might look like this. You're supposed to go A, B, C, D, and these are not genes. These are full segments of chromosomes. And this person just has a duplication there, some extra. Inversion is when it's upside down. So A, B, C, D, E, and instead it goes A, D, C, B. So this one is backwards, sort of taken up and like twisted upside down. You would think this would be no big deal because you've got all the same genes on the same chromosome, but it actually is a really big deal because the DNA is not regulated the way it normally would be. So this is a problem. And then finally, the last one is a translocation. That's when a chromosome is, it breaks off probably during a crossover event, but it doesn't go to the other one like it should. It goes to a totally different non-homologous chromosome. So this doesn't just cross over with its homolog. Instead, this little piece ends up on the wrong one. If it's a reciprocal translocation, they will be kind of shift, the non-sisters will switch with each other. Even if you end up with all the right DNA, if it's in the wrong place, it can be a problem. And that is my story. Bye.